talk about cells. We'll talk about the first part of our cell notes. Let's make this, make me a little bit smaller there. Okay. So we're talking about cell structures and their functions. Okay. So first of all, cells weren't discovered until about 1665 by a man named Robert Hooke. And he named them cells because he was looking at plant cells. He was looking at cork under a microscope, and he noticed that they seemed to be little boxes. And so it was like a prison cell. So he named them cells. Other studies were conducted by Sheldon and Schwann in the 1800s, so several years later, so a couple hundred years later, and they proposed what is now the cell theory, which is kind of the central piece to understanding what makes something living and non-living. Okay, so the cell theory has three basic parts, and it depends on what resource you look at as to whether they break it down into three parts or it's just all one statement. But I'm going to present it to you in three pieces. So first of all organisms, all living things are composed of cells. So most scientists would then say that viruses are non-living because they're not made of cells. Okay, they don't have any cellular structures. They only have DNA. So all organisms are made of cells. Cells are the smallest living things. So there are multicellular organisms like us and horses and fish, plants. There are also single-celled organisms like some of our protists, like amoebas and parameciums. There are bacteria. Those are the biggest groups of single-celled organisms. So they're still living, okay? But the cell is the smallest thing that could be considered truly alive. It does all of the life functions. And then the final piece is that cells can only come from other cells. Now, of course, the first cell had to be assembled in some way and put together, and that happened through evolution and by chance of particles coming together. But after that, we needed to have those cells give rise to other cells, which gave rise to other cells. So every cell that's alive today has come from a continuous line of other living cells. Okay. Cells are really, really small, and there's a limit to the size cells can be. Okay. They're small because they have to rely on diffusion, which is the movement of substances in and out of the cell. If you have too big of a cell, there's too much stuff in there for materials to flow in and out at the correct speed to maintain homeostasis of the cell, of the organism. That rate of diffusion, we say, is affected by the surface area available, so how much outsides there are for the cell, the temperature, things move a little bit slower when it's colder, how much concentration of stuff there is. So if we're talking about movements of water, how much water is inside the cell versus how much is outside, because that's going to change the rate at which things can move. And the distance it has to travel. And the distance that it has to travel is determined by the volume of the cell. Okay, so if a cell has a really large volume, there's a lot of material that whatever has to diffuse through has to get through. Okay, so if it's some glucose molecules, there's a lot of volume of a cell. It's going to take a long time to get through. So we want to have a smaller volume compared to lots of surface area. Okay, so we called that the surface area to volume ratio. And it's a math thing, but as a cell size increases, the volume increases more rapidly than the surface area. And I have a little diagram here to kind of show that. So we have a smaller particle, one particle that has 5 millimeter size, and you can see its surface area is 150 millimeters squared. Okay. Now, if we take that same block, that same piece, same size piece, but we break it into 8 different particles. Now our surface area is 300 millimeters. So we've increased the surface area because we've added some sides. Okay. So one thing that you can think about when you're thinking about surface area to volume ratio is think about if you have a steak. Even if you don't like to eat steak, just think about like cooking a steak. If you plop a big steak that's 8 ounces onto your grill, it's going to take a really long time for all of that heat to get all the way through to the center of the steak. Now, don't tell me that you want to eat the steak raw, because we're not talking about that. I'm talking about if it was cooked all the way through, no moo left in it. Okay, I know that's not the proper way to eat a steak, but if that's the way you eat it, then okay. So anyway, if you were then to take that same 8 ounces of steak, instead of cooking it whole, and you were to chop it up into little thin 1-inch slices, well, now that steak is going to cook in less than half of the time, because every slice that you made, you increased the surface area that was available for heat to move, and you've lowered the volume. Okay, so if we increase the volume too much, again, the cell can't move particles the way it needs to. There are cells that can overcome this size limitation by being long and skinny, like a neuron. So here's another diagram just to kind of go over that again, because this can be a kind of tough concept. So if we have a cell that has a 1 millimeter radius, okay, the volume is 4/3 pi, and the surface area is 4 pi. So that's a ratio of 3 to 0. If we have a 2 millimeter cell radius, you can see the surface area has increased, but not nearly as much as the volume. And you can look at the little water levels here, or the blue levels, to see that. And then now we've just increased that cell 1 more millimeter. And now look at how much volume there is versus surface area. So yes, there is more surface area, but look at how the ratio goes down. And we want to have a high surface area to volume ratio. Okay, so I hope that makes sense. Please ask questions if you have them, because again, I know this is not the easiest topic to go over. And then the last piece for today, for this set of notes, I wanted to talk about microscopes. So there are different kinds of microscopes, and unfortunately, with us being in distance learning, we're not able to use our microscopes, but we do have some that if we get back, we can use. So there are different scopes. Light scopes are the ones that you're going to find in most high schools and universities that you would come in contact with the most. A light microscope uses obviously a light. Okay. Now that could be a light from a lamp so you plug the microscope in and turn it on, or it could be a mirror that you turn the mirror and catch the light from outside, and those work really good too. I actually like those better than the ones you plug in. So you have a magnifying lens, and it uses the visible light spectrum to help us see or make an image bigger that we couldn't see with just our naked eye. This one over here is a compound light microscope. So this is going to be for really, really thin, incredibly thin slices of things that we're going to be able to look at. This one is a stereoscope, or binocular. You're going to be able to, you could put your finger under there and look at your fingerprints, or if you had a cut, you could look at the cut or something like that, or anything, a petri dish full of things that you just wanted to kind of see what was in there. You could look at those. Then we have electron microscopes. Well, we don't have electron microscopes because they're really super expensive. They have one at Beloit College. I got to use it one time, supervised, of course, because, again, they're very expensive. But these use a beam of electrons, and basically there's 2 kinds of electron microscopes. There's the TEMs and the SEMs. The TEMs transmit electrons straight through the material, and then the SEMs beam electrons onto the surface, so it kind of surrounds the surface. And these microscopes are the ones that produce these amazing images, like all of this pollen that you're seeing here. So you get these really detailed, beautiful images of things that are really, really, really small. And then, so as far as like, what are we going to look at for size of things? So there are some things that we can see with the naked eye, a little bit smaller than a millimeter. We can still kind of pick it out, although we're not going to see any details. Okay. Our eukaryotic cells, the cells that we have, are going to lie right in this range of 100 micrometers to 10. Okay. So we're going to need a microscope to see those, or even things that are a millimeter. We're going to use those light microscopes. And then to see things like specific, some organelles we can see with a light microscope, but to see really good details on bacteria, viruses, protein shapes, cell membranes, other smaller molecules, we're going to need to use an electron microscope to see those. Okay. So you can look up lots of cool, look up electron microscope image of, and then whatever it is you're interested in, and there's probably some Google image that is pretty cool to see. Okay, so that's it for today, and we'll come back and talk about similarities between all cells in the next session.